Hello everyone. I would like to welcome you to this year's AGBT PacBio workshop. My name is Jonas Korlach. I'm the Chief Scientific Officer at PacBio. Thank you all for joining me today. I would like to give you an update on all the latest regarding PacBio and would like to highlight a particular application area of how HiFi sequencing can be used in the area of rare and inherited diseases. However, in view of the pandemic happening around the world, I would first like to spend a few minutes on speaking about coronavirus genome sequencing. With the emergence and rapid spread of new coronavirus variants, the urgent need to better detection and tracking of these variants has been recognized and prioritized. And of course, genomic sequencing is the core method for this and for establishing a robust COVID surveillance network. We have been privileged to work with the researchers at LabCorp starting in April of last year to develop a high-throughput workflow for complete SARS-CoV-2 viral genome sequencing. LabCorp then received a contract from the CDC to apply this pipeline for a large-scale longitudinal genomic survey that aims to improve understanding of these mutations, how they are transmitted, and how the public health response to these mutations can be improved. Through this program, the CDC aims to more than double the rate at which it conducts genomic sequencing of the COVID-19 virus. Details about the sequencing pipeline are shown here, using 1.2 kilobase amplicons that span the entire viral genome. Pools of up to 900 samples per SMRT Cell are sequenced on the Sequel II or Sequel IIe systems. This effort has already delivered impactful results. For example, PacBio sequencing was used to identify the first instances of the more problematic variant from South Africa, first in South Carolina in late January, and subsequently a few days later in Maryland, Virginia, and only a few days ago relative to the time of this recording in the state of North Carolina. This highlights the power of partnering complete, high-quality viral genome sequencing with LabCorp's national network of sampling, allowing the detection and the evolution of new viral strains in time and space in the US. The pie chart shows the volume of data uploaded to the GISAID database during the week of this recording, with PacBio entries accounting for about half of all the entries. A detailed end-to-end protocol describing the PacBio HiFi viral for SARS-CoV-2 workflow is now available to allow anyone interested in implementing this real-time surveillance workflow to do so. We believe that highly accurate long reads offer critical advantages for surveillance, and I've listed 5 aspects here, which I would like to comment on in a little bit more detail. First, through the use of longer 1.2 kb amplicons, the PCR reactions are simplified while also providing more even genome coverage and striking a good balance relative to the integrity of the viral nucleic acids in the samples. Second, with fewer amplicons, primers anneal to less of the viral genome, lowering the chance that new viral mutations will cause dropouts due to primer mismatches. This is shown here in more detail. The PacBio workflow only uses 29 amplicons, corresponding to only 4.5% of the viral genome covered by primer sequences. This compares favorably to the Arctic primer set, for which almost 1/5 of the genome is covered by primer sequences, or the Ion Torrent workflow, which has priming to about 40% of the genome. So with that workflow, there's almost a 50-50 chance for a new mutation to fall into a primer region. And third, 99.9+% HiFi reads from longer amplicons allow confident detection of all variant types, from SNPs to up to 300 base pair indels, which have been seen. And they allow for phasing of variants in samples that contain multiple subtypes. This is shown by example in the next 2 slides. The first shows 2 examples of 2 different deletions of approximately 300 base pairs in length in both cases. In both of these cases, these are completely contained within the 1.2 kb amplicon. In contrast, they would interfere with the PCR of amplicons from the Arctic set. The second example shows the clear presence of 2 distinct viral subtypes, with several variants present in this approximately 2 kilobase region. Through full-length HiFi sequencing of the longer amplicons, these variants are automatically phased over long distances, thereby allowing the confident identification of two distinct subtypes. This is something that would not be possible here with the shorter Arctic amplicons. In addition, because each HiFi read is extremely accurate, coverage of only a few reads is sufficient to very confidently obtain a high-quality consensus and make these variant calls. And this also helps with pooling and amplicon balancing. From a workflow perspective, the protocol is built to allow for flexible batch sizes and can be scaled to sequence from as low as 48 samples up to 900 samples per SMRT Cell. The high level of multiplexing makes this very cost-effective, so per sample costs are very comfortably within the reimbursement rates for complete viral genome sequences. To summarize, we believe PacBio HiFi sequencing is a very powerful technology for enabling robust pathogen surveillance. And while today's efforts and focus are on detecting and tracking COVID-19 in our communities, the infrastructure that is being developed will bode well for our preparedness and sophistication of fighting COVID-19 and other pathogens in the future. So getting smarter on understanding the impact of COVID variants on vaccine responses, patient prognoses, and then longer term to quickly identify what pathogen is responsible for respiratory infection and what strain it is, whether it is bacterial, viral, something known or something new, will be possible. We are looking forward to working with you on implementing these solutions, so please reach out to us if you're interested, either at our COVID landing page shown here, or you can also use this QR code to get in touch with us. So now I would like to start with the main presentation. As I assume you know, PacBio is a leading provider of complete and accurate long-read sequencing. Through our instrumentation of the Sequel IIe and associated consumables, we provide solutions to the scientific community in all areas of genomics, whether it be genome assembly, detecting all variant types, targeted sequencing, long-read RNA sequencing or isoseq, or metagenomics. The value of PacBio data has now been demonstrated in well over 8,000 peer-reviewed publications. Most recently, the adoption and enthusiasm has centered around HiFi reads, which are sequencing reads that are both long and highly accurate. You likely know how HiFi reads are generated. In short, the polymerase reads in PacBio sequencing have become so long now, certainly well over 100 kilobases, to allow for building consensus from multiple passes on a SMRTbell template, leading to HiFi reads that are typically 15 to 20,000 bases in length and are 99.9% or greater in accuracy. On the right, I compare the read accuracy distributions of the three major sequencing technologies. And you can see that PacBio HiFi reads, very close to Illumina reads, are extremely accurate, over 99.9%. This is in contrast to other long-read sequencing technologies that are much less accurate. Secondly, the HiFi read distribution is very narrow, which makes the bioinformatics much easier and much faster because the algorithms can assume this very narrow range of high accuracies. This is also very different compared to other long-read sequencing methods. In addition to the extraordinary accuracy, HiFi reads provide other advantages, such as even coverage, high genome completeness, allele resolution, and long-range phasing. All these aspects taken together result in superior performance with respect to the calling of all variant classes. Indeed, both the recent Precision FDA Truth Challenge version 2 and the Genome in a Bottle 0.6 benchmark highlighted PacBio HiFi data having the best performance and with substantially lower variant calling errors in SNPs, indels, and SVs compared to all other sequencing technologies. One of the first areas where we believe the superior variant calling could be leveraged is in the area of rare and inherited disease. While individually rare disease cases are common in the population, with about 1 in 10 individuals affected, and it is estimated that about 80% are genetic in origin. However, currently over half of the cases remain unexplained. Similarly, out of the 8,500 known Mendelian disorders, currently about 40% have an unknown genetic cause. It is our goal this year to demonstrate that with the more comprehensive and accurate whole genome that PacBio HiFi can provide, we can demonstrate an increased resolving power. We believe that high-quality whole genome sequencing is the future of medicine and that this will be the first of many impactful demonstrations. Here's the reason why we believe this. It has been shown over the years that more complete detection yields more insights, starting with karyotyping, then microarrays, and then short-read exome or genome sequencing, resulting in increases to the now about 40% levels. So with a more complete detection of all these variant types, we can anticipate a greater explanation rate. The evaluation of PacBio HiFi data for identifying the underlying genetic cause of diseases is now in progress. And we have been very pleased to see PacBio HiFi sequencing being adopted by leading medical institutes and consortia. Shown here are examples of some of these, as well as announcements of initiatives and programs. And so what I'd like to do in the following slides is show a few examples by the researchers at these institutions explaining cases that were previously unexplained with NGS, and also highlighting the wide range of different variant types that can now be resolved with PacBio. Case number 1 was actually the very first demonstration of long-read sequencing identifying the cause of a Mendelian disease. This was the case of a 22-year-old male with recurring heart tumors that was unexplained with NGS. Ewan Ashley from Stanford applied PacBio sequencing and found a 2.2 kilobase heterozygous deletion in a gene. Ewan Ashley commented that PacBio allows to illuminate dark corners of the genome like never before. In a recent publication from Jeremy Schmutz and Greg Cooper from Hudson Alpha, they described the NGS unexplained case of a pediatric female with intellectual disability, seizures, and speech delay. Using PacBio, they found a 7 kb insertion as the underlying cause. I also want to highlight that in contrast to other techniques like BioNano, because PacBio is a sequencing method, you get all the information about the insertion, precise breakpoints, and the base resolved sequence of the inserted piece. For example, allowing here the understanding of a duplicated exon and where the inserted sequence came from elsewhere in the genome. Case number 3 is from a paper from Dr. Matsumoto from Yokohama City University, describing a now 12-year-old girl who was diagnosed with intellectual disability at 5 months old. So this was an over 10-year-long diagnostic odyssey. PacBio found a copy-neutral 12 kilobase inversion explaining this case. Also, because HiFi reads are so accurate, it allows to determine that 1, the inversion is de novo, 2, again, precise breakpoints, and 3, the inheritance, in this case from the mother, allele 1. Sometimes the underlying genetics can be very complex. This example from Kristen Zund and Matt Heastand from Cincinnati Children's of a teenager with a number of symptoms. Unexplained with NGS, they used PacBio to resolve a very complex rearrangement on chromosome 14 spanning 13 megabases with multiple inversions. The last example for a structure variation today is the case of multiple family members with seizures and ataxias, investigated by Emily Farrow, Tommy Pastina, and Neil Miller at Children's Mercy, Kansas City. I understand this was a diagnostic odyssey over many decades, which was finally explained with PacBio, finding a repeat expansion in the affected family members. Repeat expansions are notoriously difficult to resolve with NGS, whereas PacBio HiFi provides the exact links and sequence content. I would also like to show examples of detecting SNPs and indels rather than structure variants. From the same researchers is the case of a 4-year-old girl with liver problems. PacBio found 2 mutations in the same gene, and the long insert lengths and high accuracy of HiFi reads allowed to confidently call this as a compound heterozygote spanning 10,000 bases. This is very difficult or impossible with NGS, since you need the read length and long-range phasing for this determination. And lastly, here's the case of another 4-year-old girl with developmental symptoms that was just a heterozygous SNP. However, it fell into a GC-rich region of a first exon of a gene that was poorly covered despite 50-fold NGS coverage. Many first exons are GC-rich and can suffer from this problem. And while there are few NGS reads, the quality is not high enough to allow for a confident answer For example, there are no reads for the second allele, so NGS doesn't even determine the heterozygous nature of the SNP. In contrast, PacBio HiFi coverage is even and clearly shows the het SNP variant. So in summary, I hope I have given you a few examples of how PacBio HiFi sequencing has been used by the scientific community to help end the diagnostic and also the testing odyssey for these families. What is powerful is that one uniform assay, high quality whole genome sequencing, is employed, confidently and comprehensively detecting all the different variant types shown here. Therefore, it is a single genetic assay that can be used to achieve this. So coming back to this table, what we have seen so far from the collaborations, presentations, and publications to date is that more than half and perhaps up to 2/3 of the cases could be explained through PacBio HiFi sequencing. This number could increase further in the future since the analytical tools and databases, especially for structure variants, will continue to improve over the next few years. Just a very quick word about the upfront workflow. We have developed an efficient and scalable workflow which allows many samples to be processed in parallel, thus removing a potential bottleneck for the sequencing. This includes all the steps from starting with DNA as low as 5 micrograms through shearing, library prep, size selection, and sequencing, either using multi-channel pipetters or for even more samples, fluid handling automation. Similarly, there's an end-to-end data analysis workflow going from the HiFi data all the way to the visualization and interpretation of candidate variants and using public tools. The workflow is available on GitHub and will also be available soon through Microsoft Azure. One more note about data analysis. Through the introduction of the Sequel IIe system and its on-instrument processing of HiFi reads, the demands on data storage, transfer, and compute are dramatically reduced to levels of 70 to 90% reductions, and with cloud computing as an option. This results in a compute cost savings of Sequel IIe over the Sequel II system of around $700 per human genome. I would like to make one last but certainly very important point about human whole genome sequencing with HiFi data. So far, I've only spoken about the first row of this table, that is the identification of the causative variant. However, because the entire genome is sequenced with high quality, anything else that you might want to know about the germline is also immediately available. I'm showing here examples of genes important in pharmacogenomics like CYP2D6 or high-resolution HLA typing results, both of which are typically not available from short-read whole genome sequencing. Therefore, HiFi whole genome sequencing represents a single comprehensive assay that is one universal genetic assay that can encompass all others. And we think that this concept can be applied widely to many areas. I just spoke about rare disease today, but this concept of having a genome as a platform and mining this platform for information that may be needed during the different stages of a person's life and applying it for different types of disease areas, some of which are listed here, is a powerful paradigm for precision medicine. And we will continue to build on the technology we have today through improvements in all aspects of sequencing in order to make this concept of genome as a platform ever more efficient and cost-effective in the future. And that is also what our recently announced collaboration with Invitae is aimed at, to accelerate long-read whole genome sequencing into routine clinical care. This leverages PacBio's HiFi reads to jointly develop a production-scale, high-throughput genome sequencing platform, which will enable Invitae to dramatically scale their whole genome testing capabilities, and at cost substantially below $1,000, thereby opening adoption into routine medical care. In summary, I hope I've been able to share some of our excitement about what is possible with HiFi reads and the Sequel IIe system through the two examples of COVID-19 surveillance and explaining rare and inherited disease cases by providing higher quality data for better biological insights, affordable and efficient workflows, easy to handle and interpret data, and with the benefits of one technology providing all the information. With that, I will conclude. I thank you for your attention If you have any questions, please visit our website. You can also scan the QR code. We look forward to connecting with you. Please be safe and have a good day.